Hi, my name is Ethan Murdoch. I'm an undergraduate in Dr. Aaron Curry's lab in the Department of Chemistry and Henry Eyring Center for Cell and Genome Science. My poster is titled Predicting Quorum Sensing Signals Using Enzyme Co-Occurrence Analysis. And to begin, gram-negative bacteria utilize signaling molecules called acyl homoserine lactones to monitor their population densities and regulate behaviors such as biofilm formation and the production of extracellular natural products, an important subset being antibiotics. These signals are produced by acyl HSL synthases, some of which utilize coenzyme A activated compounds as substrates, as seen in Figure 1. The complicating part of this pathway is that very often these initial substrates are not produced by the strain itself. It gets it from the environment. In the literature, some of these strains grow on plants and utilize aromatic acids that the plant produces. Because of this, it's very difficult to isolate these signals in the lab because we cannot predict from the sequence of the synthase what substrate is necessary, and obviously without the substrate, no signal is produced. However, by analyzing the co-occurrence of these synthases and already characterized CoA ligases, we hypothesize that there will be statistically significant connections between synthase and ligase bins that are in the same pathway. This is to say that ligases are much better annotated than synthases, so if we can find some significant connection between groups of synthases and groups of ligases, we may be able to predict which substrate and which signal a synthase produces. The method for this is that bacterial genomes containing CoA utilizing synthases were identified on the JGI-IMG database, and their synthase and ligase sequences were downloaded. These enzymes were then grouped into isofunctional bins using sequence similarity networking, and a presence-absence matrix was created. We then performed a Fisher-Exact test of significance and calculated a risk coefficient for all co-occurring pairs of synthase and ligase bins. Currently, significant connections are being tested by growing cultures with or without the predicted substrate to look for HSL production via mass spectrometry. Special attention is being paid to co-occurring bins that bear what we call a sufficiency relationship as detailed in Figure 2. In essence, if some synthase is present only when some ligase is present, or the synthase is sufficient for the presence of the ligase, and conversely, the presence of the ligase appears necessary for the presence of the synthase, which may give confidence to a claim that they're in the same pathway. Figure 3 shows a network we created of synthase bins in green and ligase bins in blue, edges being significant connections, and those connections bearing the sufficiency relationship being shown in red. Future directions for this project include genetic experiments to determine if knocking out some synthase and or ligase stops the production of the signal. And following this, we would like to expand the method to include other pathways to these CoA-activated compounds, as in the literature, there are some examples of oxidative decarboxylation being used as opposed to a ligase. Thank you very much.